All right, so welcome to the second review session for this week, week 10. So this week we're going over DNA repair and damage mechanisms. So first we'll go over a little bit about DNA polymerase proofreading. So DNA polymerase, it's not perfect, obviously, but it's pretty good for in terms of like how little errors occur. It's about one mistake in every 10 million nucleotides added. And so there's an additional subunit with DNA exonuclease activity that allows for the proofreading function, and it moves the incorrect nucleotide to the exonuclease active site, and it cuts off, as you can see on the diagram on the right, it moves 3' to 5', its activity, which is a big reason why DNA strands are synthesized 5' to 3'. And so, as you can see here, this is the kind of the proofreading section. So this is the on the screen, this is the DNA polymerase, and here is the proofreading and or polymerizing and the exonuclease activity right here, these different subunit domains here. And just gonna keep going. But proofreading isn't perfect. Obviously, 1 in 10 billion, or sorry, 10 million is still a lot when the human genome is like about 6 billion base pairs or nucleotides long. So obviously this is going to result in many mutations, many more than that it would be way too unstable. So there's mismatch repair, which also helps reduce this rate to about 1 in a billion. So if there's 6 billion, obviously it's only 6, which is much better. And so this is done after the DNA replication occurs. So it looks for base mismatches that occur in DNA replication. And so the key point to this is that the key to this working correctly is that it needs to differentiate between the newly synthesized strand and the original parent strand. Because if it needs to know that the newly synthesized strand is the one that has the incorrect nucleotide, because the DNA polymerase has been placing down nucleotides to match up with the nucleotides on the original parent strand, and that's it's been building this newly synthesized strand. So the mistakes are in there. And so it needs to know that the newly synthesized strand has the mistakes, and if there's a mismatch, it needs to know that that's the one that needs to be fixed, not the original parent strain. And so there can be bad outcomes, obviously, if the different strands are not differentiated correctly, because if the original parent strain gets changed instead of the newly synthesized one, then it's going, every time that DNA replication occurs, there's going to be many more mutations that occur, and mismatch repairs are not going to live up to its one mistake in a billion correction. So, right, and so continue on with this incorrect identification. So if no repair occurs, the mismatched pair would remain in the DNA with one daughter cell with the mutation. As you can see here in A, if there's no repair, so the orange strands are the parent strands, or and the bottom, the bottom orange one is the template strand, and the top orange one is the newly synthesized one. But in the next round of DNA replication, if these were to be mismatched and not repaired, then one daughter cell is going to be unchanged, it'll be fine, but then the other one is going to be incorrect. And if the cell is, for this top one right here, the cell is going to think that this is correct, because it in reference to itself, it is correct because its parent strain came from a wrong parent from the previous one. And so another situation that can happen is that the second situation out of the three is that the repair occurs on the parental template strand. This will be bad for the cell because it'll end up with both daughter cells having a mutation, because if this pairing right here was supposed to be G and T, C, where the C is supposed to be paired with the G here, but instead it gets an A. In this second situation where the repair occurs on the template strand, that was correct. It's going to end up with the G being replaced to a T to match up with what the mismatch repair thinks is correct, the newly synthesized strand. And it will end up messing with both of the cells that come out of the next round of DNA replication, as they will both have the wrong pair of AT instead of GC. And then of course, the last situation is that the repair occurs in a newly synthesized strand, which is the one that we want this to occur on to have the least amount of mutations. So the cell in this case recognizes the top strand as the newly synthesized strand as the one to change out as the one with the mistake. And that would be the A nucleotide. It replaces it with a C. And so this way, next time DNA replication occurs, assuming there's no mistake there, it's going to be correctly base paired with G and C in both cases. So the mechanism of how this works is that mismatched regions of DNA these are known as lesions or distortions in the DNA because of the mismatch in the bases, and the bases have different sizes to them. If they have the wrong size, there'll be like a physical distortion in the DNA backbone, whoops. And so the organisms with this mechanism have to have some sort of a protein to sense or identify these lesions or distortions. So they have to correctly identify the newly synthesized strand as the newly synthesized strand, so that we don't have the issues that we had in the previous slide, where the parent one was recognized as the one with the incorrect pairing. And so different organisms have different ways of identifying the strands. In prokaryotes, for example, the DNA is hemimethylated after replication for a short period of time. And so what that means is that, So what that means is that the DNA has methyl groups added to it by default on the parent strain. And so when the newly synthesized strand is added to the parent's one, it for a short period of time won't contain these methyl groups. And so that's why it's called hemimethylated, the DNA, because it's only half methylated, only the parent strains have it. So when this mechanism goes to look for these lesions or distortions, it's going to scan through, and when it finds one, it's going to say, okay, there's a mismatch here. Which side has methyl groups? And the side that does not have methyl groups is the one that has the nucleotide that has the incorrect nucleotide and needs to be replaced to match the methylated parent strains nucleotides. Now in eukaryotes, it works a little differently. It's not, it hasn't been like concretely discovered, but but their hypothesis is that there is a newly synthesized that the newly synthesized strands have more nicks or in the DNA, or nicks are areas in the backbone that are disrupted. So there'll be like the nucleotides aren't connected from one nucleotide to another. They'll just like won't be connected, just like there are gaps in the DNA when the Okazaki fragments are synthesized. And so continuing on, so after the area has been identified, the area around the mismatch has to be removed. And so usually the area is removed, like there's an entire chunk that ends up removed. So there'll be a nick on the newly made DNA strand, and helicase. Sorry. And so, actually, yeah, helicase separates the template strand from the mismatch strand. And so what it does is, since there's a nick there, it starts from the nick and it will separate the strands, and the exonuclease will remove these regions. And then single-stranded DNA binding protein is recruited, as well as to like put these together, and sliding clamp and DNA polymerase are recruited again, and they will fill in the gap. And then finally, the last step is that the DNA ligase will seal the last nick, which is the space, like I said before, between the 3' of the new DNA and the 5' of the old DNA along this strand, along the newly synthesized strand. And in prokaryotes, the new DNA will also be methylated at this point. And so some common types of DNA damage and how they occur, we'll go into those now. And so the first one that we'll talk about is a base-specific DNA damage called depurination. And this is when a purine base is removed due to the spontaneous breaking of the sugar-base covalent bond. And so this results in what is called an abasic nucleotide. So it won't have a base associated with it. And this will be, this will occur on guanines or adenines. And so, yeah, so you'll have to remember the difference between purines and pyrimidines, because purines are the ones that are going to, they're the ones that are going to be, the adenine and the guanine are the ones that are going to be released during this, or removed during this breakage of the sugar base covalent bond. And this occurs about 5,000 times per day per cell, but it can vary depending on various factors, like whether a person smokes or not, and when it comes to lungs, or like getting UV radiation from tanning beds or sun, or being exposed to the sun too often without sunscreen. And then a different base-specific DNA damage that can occur is deamination. And this occurs when uracil, or cytosine, sorry, is changed to uracil. And this occurs through a chemical process here that you don't really need to know, but basically it changes the CN bond on the cytosine to a C double bond O bond on the uracil. So the important thing to remember is that it changes the cytosine with uracil, which if you remember, it's the equivalent of, uracil is the equivalent of tyrosine, or the T in DNA, but uracil is usually an RNA nucleotide. So this obviously is not good. Whoops. Because it won't be able to, obviously, it doesn't like relay the same information as a nucleotide as the cytosine that it was supposed to. And this occurs a little less, but it still occurs about 100 times a day in a cell, and again varies. And the next one that it can occur is pyrimidine dimers. And this occurs, or what this is, first of all, is it's a covalent linkage between two adjacent pyrimidines, and it usually occurs between thymines, and it'll form what's called a thymine-thymine dimer. So you can see here on the right, there's a figure that shows the 5 and 6 positions on the thymine nucleotides that are adjacent right here in the DNA backbone that form this bond. And it's usually caused by high energy wavelengths like UV lights and such. So wear sunscreen. And it can cause these distortions or lesions in DNA helix because it is forming this bond here. And next we will move on to the primary repair mechanisms for these DNA damages. So the general gist of base excision repair, which is used to repair the depurinations and deaminations we just talked about. So it varies for both of them, but they have a different first step, but then they converge in how they work. So deamination has like an extra first step where it has the identification of the uracil, and it has that bond cleaved between the uracil base and the sugar, which causes it to be an abasic nucleotide, which means it's a nucleotide without the A, T, G, or C. And so it would just be a sugar and a phosphate in the backbone, and it would have like nothing in the middle there. So once that is done for deamination, the process is the same, because deamination, the first step is to bring it to an abasic nucleotide, which is what the consequence of the depurination was in the first place. So now in both cases, you have an abasic nucleotide, and AP endonuclease and phosphodiesterase will cleave the phosphodiester bonds on either side of the abasic nucleotide. So it'll be kind of cut out of the backbone. Then sliding clamp and DNA polymerase will come and attach and fill in the missing nucleotide. And just like before, in mismatch repair, when you fill in these gaps, DNA ligase needs to come in and seal the nicks, which makes the last phosphodiester bonds between the 5' and 3' of either side of the newly synthesized strand. And here's just a graphic of how this is working. And so the next one that we'll talk about is nucleotide excision repair. So this is used to repair large lesions or distortions in the DNA, like the pyrimidine dimers we just talked about, which was the covalent bond between two T's, and it could be any two nucleotides, but it occurs most often between two T's. But in this example on the right here, it occurs between a C and a T. So the way it works, the nucleotide excision repair to fix this is to identify the lesion using a protein, And this protein has an endonuclease activity. So it nicks the DNA on either side of the lesion. It's basically the opposite of, as you saw back here, it is the opposite of DNA ligase, which in every step is used to seal the nicks, seal to form the phosphodiester bonds in the backbone. And so, in this one, yeah, like the other ones where there's endonuclease activity, endonuclease is going to nick the DNA on the other side of the lesion to break these phosphodiester bonds, which allows DNA helicase to remove the DNA that was damaged, and it'll be degraded. And so once a section around, so like on the right, on the diagram on the right here, the pyrimidine dimer is shown in green, and where nuclease, the endonuclease activity will occur is shown in these blue arrows. And so the DNA helicase will remove this section right here, where between the nicks, and it's a larger section than just the dimers, and so it'll remove a section, or the entire section around it as well, of about like 10 nucleotides or 12 nucleotides, and then the DNA polymerase will come in with sliding clamp to fill in this gap. And then, of course, like before, DNA ligase seals the nicks around, or the last nick here, after the DNA polymerase has added in the nucleotides in this strand right here. Actually, you can go back. So if there was a loss of function of this excision repair, excision repair, there would be an increase in this, in these occurrences, in the occurrences of pyrimidine dimers. So now we're going to move on to a different type of repair, which, or a different type of damage, which is double-stranded breaks. So this occurs when there is a break on both strands of a piece of double-stranded DNA. And this is caused commonly by high energy particles or wavelengths assaulting the DNA. And so, like, again, this causes the double-stranded break. And this can also be caused by unrepaired lesions or pyrimidine dimers. It can disrupt genes and cause different chromosomal rearrangements, which can end up leading to either cancer or apoptosis. And so there's a couple of different ways to repair the double-stranded breaks. There's non-homologous end joining and homology-directed repair. So non-homologous end joining is a process in which first has to identify the broken blunt ends of the DNA. So as you can see on the right here, it finds where the DNA has been broken on each strand, because remember, they can be broken. It's likely that they will be broken at different points along the strand for each one. So the first, it has to be identified in non-homologous end joining, and then it processes these ends with exonucleases. So it cuts the broken ends right here so that there can be flat ends right here and they will be joined again together through a complex of proteins. But so there's like pros to this, like obviously if you use this, you don't need a homologous strand of DNA. Any DNA double-stranded break can be quickly repaired through this mechanism. However, since you have to since you have to cut off pieces of DNA within the strand in the middle here in order to get rid of the overhanging pieces so that they can have flat ends here that can be joined by this process, there's going to be a lot of DNA here, or there's going to be some DNA here that's going to be cut out. And so there's going to be a loss of nucleotides every time this occurs, whenever this process is used. So there's going to be, as like the figure on the right says, there's going to be a deletion of nucleotides at the repair site. Okay, so homology-directed repair is a process in which it's another process used to repair these double-stranded breaks. And so this process is one in which the DNA repair template is used to repair, to perfectly repair the double-stranded break. And so this mechanism, what it does is it uses homologous recombination or breakage induced replication. So as you can see on the diagram on the right, it compares both non-homologous end joining and homology-directed repair. So in homology-directed repair, it's a bit different because it uses another template. So it can use like a sister chromatid if available. It needs a whole other copy. So when there's a whole other copy of the DNA, double-stranded DNA, it'll cut the DNA and it'll use it as a template to create or to fill in these gaps here when they're double-stranded breaks, and they can repair both sides of the damaged DNA perfectly because it has this homologous sister chromatid or externally supplied double-stranded DNA. And so this allows for the perfect, like I said, the perfect repair of a double-stranded break. However, so that is a big pro of this. There isn't the deletion of nucleotides like in non-homologous joining, but the cons, of course, is that there must be another double-stranded version of the broken DNA. And so you can imagine if the cell is in G1 just before DNA replication occurs and this happens to the cell, this double-stranded break, of course, the cell is not just going to have another sister chromosome, 2 copies of every chromosome lying around. And so the breaks in the DNA are not going to be able to be fixed using homology-directed repair, which would be ideal. And so the cell would have to use non-homologous end joining. All right, so that is the end of the review session for today. Are there any questions on the content before moving on to problem side questions?
Were you going to go over the questions that were in the PowerPoint from?
Yes, I'll go there after though.
Oh, okay.
All right, so I'll stop sharing.